you hear me okay? Thank you. I'd like to thank the selection committee and the foundation for this wonderful honor. I think this is a recognition of not only the research done in my group, but also really a recognition of the emergence of this nascent field called computational psychiatry, which combines computational neuroscience theory with psychiatry. So for many years, research in my group has been driven by the question why certain cortical areas, such as the prefrontal cortex, are more involved in cognitive functions than, say, early sensory information processing, and what are the properties that enable those kind of systems to subserve cognition. In that effort, we were very much inspired by the pioneer work of Patricia Goldman-Rakic. For example, she has done really very important work on a very basic and fundamental cognitive function called working memory. So working memory is our ability, as you all know, to hold and manipulate information in the absence of external stimulation. So the neuron process underlying working memory representation must be internally maintained. It cannot be driven by external stimulation. In this particular beautiful spatial working memory paradigm, they found that single neurons in the prefrontal cortex show this self-sustained so-called persistent activity that encode particular information when a subject, in this case a monkey, is holding that information in mind across a delay period. So the question, really impressed by this beautiful experiment is how does the circuit do it, right? In fact, this kind of persistent activity has been observed in multiple brain regions. That's as indicated here by this, in this review. Those are the areas that commonly show persistent activity. And those are typically the areas in the prefrontal cortex, posterior parietal cortex, and inferior temporal cortex. The general idea is that those areas are endowed with strong enough recurrent synaptic excitation. And that enables those kind of circuits to sustain activity without external drive. Okay, that free the system from the immediate environment, and so you can become more flexible. You can control your behavior, you can think, you can hold and manipulate information in your mind. So we decided to test this idea quantitatively using what we call biophysically-based or biophysically realistic neural circuit mathematical modeling. So on computer, we build mathematical models that are really based on two major pillars of neuroscience, namely neuron physiology that describes synapses, neurons, in terms of their cell biology and biophysics. On the other hand, the wiring diagram that quantifies connectivity, right? Using those two, lines of information, you can build a mathematical model that accurately captures a circuit to some degree. And that ended up with being mathematically a so-called nonlinear dynamical system that you can be used as a platform to examine, for example, the idea that if you parametrically change the strength of recurrent excitation, do you start to see the emergence of working memory representation? Here's an example of our actually theoretical model that shows indeed that if you gradually change the strength of recurrent excitation as a drive, the workhorse for reverberation in the circuit, then at some point there's a threshold. There's this emergence of memory states. So as schematically shown here, those dots represent a family of self-sustained activity states. I'm going to show you an example in the next slide. So that is really a very important, interesting insight from theory that even some modest changes, graded changes, can lead to qualitatively different behavior without persistent activity over here. Maybe early sensory areas like V1 sits over here. That's why they don't show working memory representation, with an area like prefrontal cortex may sit over here with just enough recurrent excitation to self-sustain persistent activity. All right, here's an example of that kind. What's shown here is the spatial temporal pattern of such a neural circuit model of prefrontal cortex. So a brief input activates a group of neurons that, through this reverberation, can sustain the activity throughout the delay period in yellow, right? So at the end of the delay, you can read out the remembered cue by looking at the peak location of this bell-shaped activity pattern that's self-sustained, right? The precision of working memory representation is quantified by the width of this bell-shaped activity pattern. All right, that's going to be important when I talk about how we apply this kind of model to schizophrenia. It turns out that the same model we used can also account for decision-making computation, which was a total surprise to us. Without going into details, just to say that when in the experiment, you gradually manipulate the task difficulty in a decision task where you evaluate different options leading to a categorical choice. So from right to left, you increase the task difficulty, two things happen. Namely, the performance, you do worse when the task of your decision-making gets more and more difficult. And secondly, the time it takes for you to decide gets longer and longer. So the reaction time is longer, okay? And what happened when you try to make a really hard decision that takes a long time is that neurons in your brain show this transient ramping activity that allows the brain to accumulate information for or against the different options. So our model, this is a model simulation showing how this kind of ramping activity actually takes place, and that has also been observed in single neurophysiology of behaving animals. So the model has this interesting duality. On one hand, it can show this time integration of information in the form of stochastic ramping activity over a rather long period of time, comparable to the reaction time. On the other hand, it also shows this attractor dynamics leading to a categorical choice. It's the same mechanism that can also sustain the activity when the input is gone in working memory. Okay, so this is an interesting emerging idea. This is a shared mechanism that can do working memory and decision making, and the key for that is slow reverberation. That depends on NMDA receptors. It turns out that our kind of model really does not work without having this NMDA-dependent slow reverberation that can explain working memory and decision making. So that specific prediction has been tested in a very elegant experiment in Amy Einstein's lab in the monkey experiment. And that's very interesting because the combination of model prediction and monkey experiment, no human experiment, ties very well with the known evidence from human studies, from John Crystal and others, suggesting that NMDA receptors are important for understanding prefrontal function. Okay, so this gives you a very specific example why the kind of model we build could, in principle, start to make a connection with psychiatry. So those are the kind of examples that I think, by our work and others, led to this emergence of so-called computational psychiatry. So let me give you a case study about how we really can use this kind of model to look at the deficits, let's say, associated with schizophrenia. So there's some evidence that NMDA hyperfunction is a very major characteristic of schizophrenia. And in fact, the modulators like dopamine act on those kind of systems, right? Either excitation or inhibition that I'm going to talk to you about a bit later. But if you dive into more details, the NMDA hyperfunction can act in two different ways. Either you have a reduced excitation, this is reverberation, right? Along this axis, if you have too much impaired recurrent excitation, then you lose working memory function altogether. On the other hand, if you go along the y-axis, you could have impaired NMDA-dependent drive of interneurons, inhibition. Okay? People believe actually ketamine primarily affects this way. Now, if you go this way, you don't lose working memory. You still have working memory representation, but you become more distractible, right? And that's quantified here because the color actually is indicating this width of the bell-shaped activity pattern I told you about, indicating the inverse of the precision of working memory representation, which is reproduced in this slide. So in the control case, you have this precision for working memory representation with reduced drive to interneurons, let's say under ketamine, then you have broader, bound attractor, okay? Now, if we learn something like this precise, this is concrete, right? You could now perhaps be inspired to think about how to repair the system, right? So in this particular model, we proposed a couple of different ways in purple and green, showing that the impaired, widened working memory representation actually can be restored back to control, if you do A or B, right? This, of course, is just a toy model, but I hope this provides an example, for the future that maybe at least the model-inspired or even model-aided understanding of exactly what goes wrong could lead to new ideas of treatment. So more recently, my group has expanded our research to look at large-scale multi-regional brain system, not just local circuit. Let me share with you a few results we have obtained so far, leading to, I think, a very promising general principle about large-scale brain system organization. So we used newly published directed weighted connectivity data from Henry Kennedy's group in France, and we built a dynamic model of the large-scale cortical circuit of macaque cortex, again, no human primate system. And one thing that just emerges in this kind of dynamical system is a hierarchy of time constants. So as shown here in response to a brief input from the visual pathway, early sensory areas show rather fast responses with, say, prefrontal cortex and some higher areas have this ability, again, of ramping activity, integrating information that's required for decision-making, and this after discharges, sustained activity even when input is gone, representing short-term memory trace, right? So you have this working memory and decision-making aspects of dynamics that emerges in this kind of system, only selectively for certain association areas. And the key for this to happen is that certain properties systematically vary from area to area, what I call macroscopic gradient, as shown as in this example. So what's shown here is the count of spines, which are the sites of excitatory synapses in a single neuron, and that can be thought of as the strength of excitation per neuron, okay? And that turns out to change rather systematically from area to area, from primary visual cortex to higher areas and up to prefrontal cortex along the hierarchy, right? So the area-to-area variations of circuit properties or cellular properties, even genetic properties, may not be random. This is a macroscopic gradient, as shown here for excitation. We found similar kind of macroscopic gradient for inhibition. I'm going to skip some details just to say that there are different types of interneurons that, by the way, are implicated in psychiatry. And there's a particular type of GABAergic cells that control the output of pyramidal cells. And there's another second type in blue here that controls the inputs onto the pyramidal cell dendrites. So that can gate input flow of the system. And what's shown here is the ratio of input controlling versus output controlling interneurons. And you see this systematic change along the cortical mantle, right? So this kind of regular systematic macroscopic gradient of inhibited neurons is quite interesting and newly reported only last year. we have a story about how this kind of motif involving different kind of interneurons can gate information throughout a very complicated cortical system. So maybe certain cognitive deficits, the reason that unfortunately certain patient groups cannot organize thought, cannot really manage organized behavior, cannot control pluripotent automatic responses, is because of the impairment of this routing mechanism. We then used such a computational model with macroscopic gradient to study distributed cognitive processes, such as working memory. So there's an increasing recognition that working memory representation involves multiple brain regions. We would like to understand how that works, how that happens, what's the underlying mechanism. This is an example of model simulation now of large-scale multi-regional cortical circuit model of the macaque cortex. You see that in a delayed response task, simulated delayed response task, the response to external stimulus is confined in the posterior part of the cortex, whereas during the delay period, when the input is gone and you're required to hold the information in mind, it's more anterior parts of the cortex, association areas, but distributed across multiple brain regions that talk to each other through long-distance loops to maintain working memory representation. We also worked with psychiatrists to apply this kind of approach to human large-scale brain modeling. And this is an example from John Murray, who is a former graduate student, now a faculty in psychiatry at Yale. He and others have built a similar type of computational platform for human cortex, combining connectivity data like diffusion tensor imaging data with functional connectivity data. So they asked recently, inspired by what I told you, whether macroscopic gradients play a role to explain the functional connectivity data from human. And what they found basically is that fitting the data with the model, the fit is much better with macroscopic gradient. The correlation between the data and the model result is 0.75 with the presence of macroscopic gradient, much better than if you assume that there's no macroscopic gradient. In this case, the correlation would be reduced to more like 0.46. All right. Now, what's the evidence for such a macroscopic gradient in human cortex? Recently, David Lewis published a very interesting paper using transcriptome data. And they found that, with a measure of what they call composite glutamate, signaling or GABA signaling, they found that there's an increasing gradient of excitation, if you like, and a decreasing gradient of inhibition. Now, they did the same transcriptome analysis of cortical tissue post-mortem from schizophrenic patients. They found that in open bars, that the excitatory gradient is blunted, basically disappears in schizophrenia, whereas the gradient of inhibition becomes more enhanced compared to control. Right? This is a very new data suggesting, indeed, this is one way to think about deficits or biological abnormality across the whole cortex. Now, finally, I want to just share with you this joint work with John Crystal's lab and also with Alan Antisevic at Yale. We tested the idea whether this macroscopic gradient can explain certain paradoxical effect. Namely, if some biological abnormality at the molecular level is common, global, in the whole brain, why sometimes functionally you see really abnormal functional dynamics or function only selectively for certain areas such as prefrontal cortex, right? What's shown here is the result of imaging data from more than 100 subjects. You see that by some measure of connectivity, actually in schizophrenia, the global connectivity is enhanced compared to control. But that happens only selectively for certain frontal areas, but not for sensory areas. Okay? In spite of the fact that maybe the underlying biological alternation at the molecular level is global, right? And we showed by computational modeling that this cannot be explained without macroscopic gradient. I hope this gives you a couple of examples illustrating how if you can combine rigorous behavioral task paradigm design, large-scale population studies, and computational modeling, you could really tease out certain details about what exactly goes wrong and what is the underlying circuit mechanism. That's basically the physiology, the methodology of computational psychiatry that I'm really grateful that the foundation recognized as an emerging field and hopefully will support in the future. Let me end by thanking people who did the work and the collaborations. Thank you.